Everyone at this international webinar, which is related to cancer stem cells. And it's truly international, as we have participants from all over the world. And hopefully it will be interesting for quite a few people. So I would like to start with an introduction. And this introduction is related to the potential role of cancer stem cells and their role in cancer chemotherapy, which, as to our understanding, chemotherapy abolishes the bulk of proliferating tumor cells. However, successful chemotherapy even leaves some cells from which cancer can relapse, and even with higher invasiveness and chemoresistance. And there is accumulating evidence that these so-called cancer stem cells are responsible for cancer chemoresistance, relapse, and metastasis. And these so-called cancer stem cells have the ability to self-renew and differentiate into heterogeneous lineages of cancer cells, especially in response to chemotherapeutic agents. There is an alternative proposal which suggests that there are no real cancer stem cells, but there are drug-tolerant persister cells with special features among the cancer cells, and this so-called DTP cell population may be the source of the cancer relapse. And therefore, it would be very important to explore the basic biology and clinical features in cancer therapy, including targeted therapies. And since cancer stem cells or DTP cells are considered as root of primary and acquired therapy resistance, this should be an important question and discussed in the present webinar. I'm trying to get my next slide, which is not really working here. Yeah, so after this short introduction, I would like to talk about the selection and targeting of cancer stem cells. The first introduction of this cancer stem cell hypothesis was by John Dick at the Ontario Cancer Institute in Toronto in 1994. So it's rather early. And he had several papers with this hypothesis and repeating this idea, which says that cancer stem cells comprise a unique subpopulation of neoplastic cells within tumors that is highly tumorigenic and relatively resistant to standard therapy. So again, a subpopulation with special tumorigenic and resistance capacities. So what do we call cancer stem cells? So these are self-replicating cells within tumors capable of regenerating the given malignancy. So it's focused on one single malignancy, characterized by a relatively low number, but this very low number, even one cell, is capable to regenerate the tumor. Moreover, these cells can produce tumor colonies both in vitro and in vivo, and show chemotherapy and radiation resistance, which involves slow division, drug transporters, mutant enzymes, higher mutation rate, etc. Well, that's a very complex idea, and I will try to show that some of these issues are contradictory. But what's the idea how tumor stem cells are generating? So one possibility that these tumor-generating mutations occur in normal stem or progenitor cells, or in the developing tumor, some cells regain stem cell properties. So the two ideas are shown here. So how cancer stem cells are generated. Are they generated from stem cells? So there are even pluripotent and other kinds of stem cells, and they are just dividing and producing again normal stem cells. But some of them are mutated and go into a tumor-like proliferation, and they generate the cancer stem cells. Another possibility that tissue-derived progenitor cells are dividing, and they are mutated and become cancer cells, and again become the cancer stem cells. But it's even possible that differentiated cells can be de-differentiated, and self-renewal would occur, and again they would produce the cancer stem cells. This latter idea is supported that by now we know that all differentiated cells can be reprogrammed into stem cells. So the next slide, which comes very slowly, but hopefully it will come. It's not working now again. Somehow I have to stop here because this is now not really working. Okay, this is our next slide. So then how do we imagine that cancer can be eradicated? So we can have the tumor with the so-called cancer stem cells. And if we unspecifically target all the tumor cells, that the tumor will shrink, but the self-renewing cells will remain alive and a relapse will occur. Now, if we can target the cancer stem cells, they will be killed or disappear, and the tumor will shrink and regress and eventually disappear. Now, it's interesting that at the same time, cancer stem cells are believed to be resistant to chemotherapy. So what happens if you use a chemotherapy, which just generally kills the cells this way, but the cancer cells, the stem cells survive, then there is a tumor relapse. If you manage to kill the cancer stem cells, there is tumor regression and disappearance. So what could be the role of the multi-drug ABC transporters in this process? I would like to focus on this area of cancer stem cells because chemotherapeutics are the most effective treatments for metastatic tumors. However, the ability of cancer cells to become simultaneously resistant to different drugs is a major barrier to successful chemotherapy. And these multi-drug transporters are able to generate such a drug resistance. So what are these ABC multi-drug resistance? They are present in our normal cells, but they can also be present in tumor cells, and they can potentially be regulated by drugs, hormones, and xenobiotics. So the major role of these ABC transporters that hydrophobic drugs, xenobiotics, or hormones can normally enter the cells through the plasma membrane, but several ABC transporters are able to extrude these drugs from the cells, and the key players are the ABCB1, ABCC1, and the ABCG2 transporters. And whenever these hydrophobic agents enter the cells, they can regulate the expression of these transporters by transcription, translation, processing, trafficking, or destruction. So in addition to their presence, they can be regulated by complex ways. Now, the key multidrug transporters, ABC multidrug transporters, are the ABCB1, the ABCC1, and the ABCG2 transporters. The ABCB1 protein can transport all kinds of hydrophobic drugs, and the C1 and the G2 transporters can transport both hydrophobic drugs and conjugated metabolites. So these three major transporters can handle thousands of substrates, and many of them are actually anti-cancer drugs. And these can be transported with an overlapping specificity by the MDR1, MRP1, or the ABCG2 protein. So whenever you use a chemotherapeutic drug, you can expect that it will be transported by one or the other of these multidrug transporters. And although you can achieve successful treatment of cancer with targeted inhibitors, usually resistance also develops. And one of the key causes of this drug resistance is the presence of the multidrug transporters. So when these transporters are present in tumors, it's interesting that during their discovery, in drug-selected cancer cell lines, there were complex gene rearrangements, gene amplifications, multiple chromosomal copies, but it has turned out that it's not the real world. It's almost only occurs in the laboratory. In the clinical cancer drug resistance, the MDR ABC transporters are also important, but they have only low levels, either ABCB or ABCG2 transporters. Very low levels can cause already a drug resistance, and there is no mutation, no gene amplification in the clinical tumor samples. So what can be the background of this defense mechanism? Selection of MDR ABC expressing cells or induction or regulation of these transporters in the tumor stem cells. And this is just a slide showing that if you have in a given cancer MDR positive cells, usually the survival is much less than without the presence of these proteins. So how they are related to stem cells? It has been first shown in hematopoietic stem cells that there is a so-called site population. So if these cells are incubated with a host dye, some of the cells actually extrude this dye, and these are called the site population cells. And in the site population, if you separate them, then the major protein which is present is the ABCG2 transporter. So it's highly expressed in the site population and practically not expressed in the non-site population cells. So how does it relate to the tumor cells? Well, there are many examples now that the site population is also present in the tumor cells. And this is again related to the high-level expression of the ABCG2 protein. And if you inhibit this protein, then the site population disappears. So it's a functional protein which is present, at least in some tumors in the stem cell population related to the site population. So how does this ABCG transporter relate to cancer stem cell resistance? So if it's present in cancer stem cells, although the physiological expression are in tissue barriers, stem cell, normal stem cells, and it's related to the removal of metabolized xenobiotics and protection of stem cells, if it's expressed in cancer stem cells, then it protects the tumor cells, and because it shows a promiscuous recognition of various drugs and xenobiotics, It shows an MDR phenotype. So one idea would be to eradicate the cancer stem cells, and that we called in some of our papers a stem kill approach, which means that you want to generate anti-cancer agents, whether they are targeted or general chemotherapeutic agents, which show selective ABCG2 interactions. So if they are transported by ABCG2, they become ineffective agents, especially in the cancer stem cells. If you generate anti-cancer agents which can evade ABCG2, that is, they are not substrates of this transporter, they are able to kill these cancer stem cells. Another possibility, effectively inhibit ABCG2 by some of the anti-cancer agents. And in this case, in addition to an anti-cancer action, that will inhibit the transporter and even promote the action of other ABCG substrate cytotoxic agents. So what could be the clinical relevance of the MDR-ABC transporters? So during cancer development, whether it comes from stem cells or already differentiated cells, some of the cancer cells, we can call them cancer stem cells, have expression, relatively high level expression of these MDR proteins, especially ABCG2. And during transformation, mutation, induction, they undergo partial differentiation and they lose this transporter expression. So when you are using chemotherapy, then you cannot kill these cells which have MDR expression, and you can kill all the other cells which do not have this. And these cells can be selected either based on mutations, induction of the proteins, or just selection. And when you have the relapse or the metastasis, already these cancer stem cells containing the drug transporters are appearing. So actually, this is an induction of cancer stem cell growth by chemotherapy. Now I will finish my presentation with the idea which are shared by many clinicians. Then the question is how to eradicate cancer and the cancer stem cells. And among gardeners, there are quite a few who think that you are able to eliminate the weeds only if you dig them up by the roots. So if you remove them one by one, then you can eliminate the weeds. But there is another general idea that it may be enough if you want to have a nice garden. It's enough just to mow the lawn frequently, and the grass will survive and the weeds will practically disappear. This idea is related to the transformation of cancer to a chronic disease. So if you can transform cancer growth to a chronic disease and treat them sequentially at the good times, then you can practically eliminate all the bad aspects of the cancer. So I will stop sharing here.